So welcome back. In this section, we'll be talking about genetic variation and mutation. Let's come back to our first idea of cancer as a genetic disease. Cancer is caused by an accumulation of detrimental variation in the genome. So what is that detrimental variation in the first place, and where does it come from? It's important to remember that most genetic variation is benign, and really it's a very good thing to have in a population. The vast majority of our genomes are the same person to person, about 99.5%. That remainder, 0.5%, is variable in the human population. This variation is very small. Only one or a few bases of the DNA vary at any particular location, and those are known as polymorphisms, or single nucleotide polymorphisms, also called SNPs. The variation in our genomes accounts for our individuality. It describes why we look different from each other, why we look slightly different from our parents or children, and no one shares the exact copy of your DNA. Even your alleles may differ from each other. One variant on your maternal allele and a different variant on your paternal allele. So then what's a mutation? A mutation is one of the few detrimental genetic variation that increases risk to developing a disease, or in rare diseases, actually causes the disease itself. When someone says that they have a cancer gene, what they actually mean is that they have a mutation in a gene that can increase cancer risk. There are two different types of so-called cancer genes, oncogenes and tumor suppressors. Oncogenes promote cancer, so in a normal healthy cell, expression of oncogenes is low, while in a cancer cell, expression of oncogenes is high. This is in contrast to tumor suppressors, which protect against cancer. So in normal cells, you'd have high expression of tumor suppressors, and in cancer cells, you would have low expression of tumor suppressors. Let's go back to our analogy of cars driving on a road. To prevent a crash, you need to both accelerate with the gas or stop with the brakes, but a balance of the two is important. This is true in normal cells. So in this case, the gas is oncogenes and the brakes are tumor suppressors. And in cancer cells, these are both misregulated. There are two types of mutation. Activating mutation causes the gene to be expressed at the wrong time, at the wrong level, or even with a new function. So this means that activating mutations in oncogenes are tumorigenic. Their converse are inactivating mutations. And these cause reduced function of a gene, so either less RNA or protein is made. And inactivating mutations in tumor suppressors are tumorigenic. So we'll go ahead and take a break now, and when we come back, we'll talk about two-hit hypothesis and genomic instability.